Hello everyone, myself Dr. Vinita Rathod, Associate Professor of Zoology at SMB Government PG College, Nandwara. Today I am discussing accessory digestive organs. In this part 1, I will discuss about anatomy and histology of liver. First, we must know that what are accessory digestive organs. Accessory digestive organs are the organs which do not form gastrointestinal tract but can develop as outgrowth and assist gastrointestinal tract in digestion of food. Various accessory digestive organs are teeth, salivary glands, liver, and pancreas. So, in this part 1, we will discuss about anatomy and histology of liver. The liver. Liver is an accessory digestive organ which is located in the upper right quadrant of the abdomen. It is cone-shaped in structure and it is dark reddish brown in color. It weighs around 3 pounds or you can say 1.5 kgs in weight. It is the second largest organ in the human body after skin. It is the largest gland in the human body. Liver performs a wide range of functions such as synthesis of bile, glycogen storage, clotting factor production, etc. Liver can perform both the endocrine and exocrine functions. Endocrine functions means it acts as endocrine glands and can secrete some secretions which are just like hormones. So, as endocrine gland, liver secrete IGF-1, which is called as insulin-like growth factor 1, which has insulin-like effects as it can bind to the insulin receptors and also act as stimulus for the growth in human body. As exocrine gland, it secretes bile which performs various functions. Division of liver. Anatomically, the liver is composed of 4 incompletely separated lobes supported by 2 ligaments. The 4 anatomical lobes of liver are right lobe, left lobe, caudate lobe, and quadrate. The right lobe is located on the right side of the visceral surface and it is 6 times larger in the size to that of left lobe. Left lobe is located on the left side of visceral surface and it is smaller than the right lobe. The third lobe is caudate lobe, which is situated upon posterior superior surface of the right lobe of liver. The last and the fourth lobe is called quadrate lobe, which is situated on the lower surface of the medial segment of left lobe of liver. There are 2 types of ligaments attached with the liver. These are falciform ligament and round ligament. The falciform ligament and round ligaments are the ligaments which are meant for the attachment of the liver to the surrounding structures. These ligaments are formed by a double layer of peritoneum. The falciform ligament and the round ligament, which is also called as ligamentum teres hepatis. These are formed by the peritoneum and they attach the liver with that of the visceral organs. This picture shows the anatomy of liver. Here we can see right lobe and left lobe. In between them, inferior vena cava can be seen. Here we can also see falciform ligament and round ligament of liver, and a part of gallbladder. The right lobe, as we can see in this picture, is bigger in size than that of the left lobe. The falciform ligament is the ligament which connects or attaches the anterior surface of the liver to the anterior abdominal wall. It forms a natural anatomical division between the left and right lobes of the liver. The second ligament, which is the round ligament of the liver, it is the remnant of umbilical vein that exists in the edge of falciform ligament. It divides the left part of the liver into medial and lateral sections. Histology of liver. Histologically, we can see following structures in the liver. First, parenchyma, connective tissue or the stroma, sinusoidal capillaries or sinusoids, and perisinusoidal space or spaces of DISSE. The first, parenchyma. The liver parenchymal cells are also called as hepatocytes. These are organized plates of hepatocytes. The hepatocytes are polygonal cells with a central nucleus. They are arranged in plates one cell thick with sinusoid on either side with a radial arrangement in the center of the lobule. Hepatocytes carry out synthetic and metabolic functions of the liver. The second is stroma, which is the connective tissue of the liver. Connective tissue or the stroma continues with the fibrous capsule of glycerol. It contains blood vessels, nerves, lymphatic vessels, and bile ducts. The third structure is called as sinusoids or the sinusoidal capillaries. Sinusoids are vascular channels between the plates of hepatocytes, and they receive blood from the terminal branches of hepatic artery and portal vein at the periphery of lobules, and they deliver it into the central vein. Sinusoids do not have basement membrane. The last part is spaces of DISSE. Spaces of DISSE lies between the sinusoidal endothelium and the hepatocytes. It contains blood plasma. The microvilli of hepatocytes extend into this space. Perisinusoidal space or a space of DISSE lies between the basal surface of hepatocytes and the endothelial surface of Kupffer cells lining the sinusoids. The sinusoids contain thin continuous endothelium with large fenestrae. Lid formation takes place in this space of DISSE. At the same time, exchange area for liver and also secretion other than bile can also be performed by this space. There are present eto cells which are also called as stellate cells. They are present in the liver mesenchyma. These eto cells are the main place of vitamin A storage by the liver. This diagram shows the histology of liver. Here we can see hepatic lobules, hepatic sinusoids, hepatic cords, central vein, and bile canaliculi. The liver contains thousands of hepatic lobules, which are the structural and functional units of liver. These lobules contain liver cells, which are called as hepatocytes. Liver lobules. Liver lobules forms the chief mass of the hepatic substance of the liver. Liver lobules are hexagonal in shape, and at each 6 corners there is a portal triad of vessels consisting of a portal vein, a hepatic artery, and bile duct. There are three different types of liver lobules present in the liver, as can be seen in the diagrams here. The first is classic lobule, the second is portal lobule, and third is called liver acinus. Classic and portal lobules. First is the classic lobule. The classic lobules consist of roughly hexagonal plates of hepatocytes. They are also called as hepatic lobules. One portal canal is located at each corner of hexagonal classic lobule, making a total of 6 for each lobule. At angles, the portal area is occupied by portal triads, which are surrounded by loose stromal connective tissue. At the central part, the central vein is present and the hepatocytes radiates outwards and arrange into strips similar to the spokes of a cartwheel. The second is portal lobule, which is roughly triangular in shape, having a central axis through portal field. The outer margins of the portal lobules are imaginary lines drawn between the three central veins that are closest to the portal triads. The portal lobule explains the exocrine bile-secreting functions of the liver. The portal lobules highlights the bile-synthetic functional unit of liver, as all the bile synthesized by the hepatocytes within the portal lobule drains to a common bile duct tube. The third type of the liver lobules are called liver acinus. The liver acinus are diamond-shaped in structure. They are provided with two axes, short axis and long axis. The short axis of the acinus is the terminal branches of the portal triad that lie along the shared border of two classic lobules. The long axis is an imaginary line drawn between the two adjacent central veins closest to the short axis. The liver acinus explains blood perfusion, metabolic activity, oxygen supply, and liver pathologies. Based on acinus conceptualization, the pathologists have designated three different zones corresponding to different degrees of oxygenation. Each half of the liver acinus is now divided into three different zones: Zone 1, Zone 2, and Zone 3. Zone 1 is the closest to the short axis. It is having highest amount of oxygen. It has earliest contact with oxygen-rich and potentially toxin-rich blood. Zone 2 is present between the Zone 1 and Zone 3, and it is oxygen intermediate zone, and potentially toxin intermediate blood comes after the Zone 1. Zone 3 is the last zone, which is farthest from the short axis, and oxygen poor and lowest in oxygen concentration, that blood comes in contact with the zone 3. So this is the zone with poorest amount of oxygen in it. Functions of the liver. Liver produces bile. Bile is an alkaline greenish fluid that breaks down the fats into small droplets to assist in their chemical digestion. Emulsification of fats takes place here with the help of bile, which reduces the surface tension and helps in the digestion of fat. Bile neutralizes gastric acids which are present in small intestine. It detoxifies drugs, metabolites, and various toxins produced in the body. It stores excess amount of nutrients and vitamins like vitamin A, vitamin D, and vitamin K and release them as and when they are needed. Liver synthesizes blood plasma proteins such as albumins, globulins, and proteins which are required for blood clotting, that is prothrombin and fibrinogen. Liver also helps in phagocytization of the debris present in the blood. It helps in the breakdown and recycling of the components of aged erythrocytes and damaged or worn out formed elements. Liver also helps in the excretion of bilirubin, which comes from the recycled erythrocytes. The biliary apparatus. It collects bile from the liver, stores in gallbladder, and transmits it to the second part of duodenum. The biliary apparatus consists of a network of thin ducts that carry bile from the liver and gallbladder to the duodenum. The left and right lobe of the liver drains bile into the left and right hepatic ducts respectively. The left and right hepatic ducts merge to form a single common hepatic duct. The cystic duct attaches to the common hepatic duct and carries the bile to and from the gallbladder. Gallbladder. Gallbladder concentrates bile which is produced by the liver and stores this bile and concentrate it until it is needed for the process of digestion. The cystic duct connects the gallbladder to the common bile duct. The gallbladder can hold approximately 40 to 60 milliliters of concentrated bile. This is the diagram which shows the connections of liver, gallbladder and pancreas with the duodenum, or you can say that the biliary apparatus. Here we can see left and right two hepatic ducts which unite together to form a common hepatic duct. The common hepatic duct then connects with the cystic duct which is coming from the gallbladder and forms a common bile duct. The common bile duct then from the pancreatic duct connects to and it opens into the main pancreatic duct, hepatopancreatic ampulla, major duodenum, duodenal papilla, and then they pour their secretions in the duodenum. So left and right hepatic ducts merge to form a common hepatic duct. Common hepatic and cystic ducts merge to form a common bile duct. Pancreatic duct merge with the common bile duct at the hepatopancreatic ampulla, and the fourth is the bile and pancreatic juices enters in the duodenum at the major duodenal papilla for the process of digestion in small intestine. So this is all about the first accessory digestive organ, liver. In the next video, we will discuss about the next accessory digestive organ, pancreas.